So good evening, ladies and gentlemen. I would like to welcome you to the Learn from the Legends series, an international bi-monthly webinar series organized by NNF Kerala and IAP NeoChap. Today we have a legend from Spain to enlighten us on a great topic. It is oxygenation of the fetus and the newborn. And the speaker is none other than Professor Máximo Vento, the Chief of Division of Neonatology, University and Polytechnic Hospital, La Fe, Valencia, Spain, and the Scientific Director, Health Research Institute, La Fe, Valencia, Spain. And today, our moderators will be two eminent neonatologists of India, Dr. Hariram and Dr. Nalini Kant Panigra. So it's going to be a great scientific feast for all those who are attending this session from all over the globe. I welcome Professor Máximo Vento to start his proceedings after the introduction by Dr. V.C. Manoj. Over to you, Manoj. Thank you.
Thank you, sir. Hello, everyone. Hearty welcome to another session of our Learn from the Legends. And today we are so proud to present you one of the greatest legends of neonatology we have, the Professor Máximo Vento. All of us have started learning neonatology hearing this name, this great name. He has been introduced as a professor of pediatrics, chief of division of neonatology at University and Polytechnic Hospital, Spain. I would go much beyond that. He is a true legend in all the words, always. He is the chairman of the European Board of Neonatology, member of the Executive Committee of the European Society for Pediatric Research, ex-president of the Spanish Neonatal Society, chairman of the Spanish Maternal, Neonatal and Developmental Network, chairman of the Platform for the Development of Clinical Trials in the Health Research Institute, La Fe, Valencia, Spain. I hope I'm pronouncing it right. And the designations go on and on. He has a very lines of interest right from the topic that we are going to hear today. His line of interest stretches all over, and I'm really amazed to, and really I feel fortunate to introduce this legend to this August gathering. Physiology of the fetal to neonatal transmission. His research runs perinatal asphyxia, resuscitation, the delivery room, oxygen physiology, hypoxia, hyperoxia, derived toxicity, oxidative nitrosative stress, biomarkers, epigenetics and oxygen, redox regulation, microbiome, infection, genome wide expression, methodology, various aspects like targeted and untargeted metabolomics, HPLC or GC coupled to mass spectrometry, LC, QT, OF approach, capillary electrophoresis. His areas of research where he has done commendable work go on and on. So, and he has a large amount of published literature in the international peer-reviewed journals. So, if I go on like this, I will be standing between you and the great legend. So, let me wind up the introduction and proudly introduce the one of the greatest legends of today, Professor Máximo Vento. Over to you, Professor Max.
Thank you very much for this, I would say, excessive introduction that you have done. I am very proud and very happy to be here with all of you. And I'm very, very thankful to the organizers for inviting me to spend this time with you. I am going to share my screen and start with my presentation. I hope all of you can hear me and you can see the presentation.
Yes, sir.
Okay, so we are going to talk today about oxygen, the fetus and newborn, a comprehensive approach. First of all, I would like to devote some minutes to oxygen metabolism and oxidative stress, because the relevance of oxygen, it regards its metabolism as a supplier of high energy to multicellular organisms. But on the contrary, it has also a drawback with the oxidative stress that it causes, that can cause damage at any age, but especially in preterm infants. Oxygen metabolism and oxidative stress and nitrogen stress will be my first approach. Then damage to DNA, epidemiologic association, and oxygen load. So would you know, and all of you know from basic biochemistry, that aerobic metabolism is 18 to 20 times more energy efficient than anaerobic metabolism. Hence, if we're breathing air, which contains 21% oxygen, every mole of glucose that we are ingesting, it's going to wind up creating 36 moles of ATP. However, if you are taking 1 mole of glucose and you would be without oxygen in an anaerobic metabolism, then you would only put forward 4 moles of ATP. Multicellular organisms, like the human body, require a great amount of energy for growth and development. And the amount of energy required is, and this is very important, tissue dependent. Not all the tissues behave the same. For example, the brain. The brain has a huge metabolic rate. It has a high transmembrane potential. Most of the energy that the brain receives is spent keeping the high transmembrane potential in order to keep sodium outside the neurons and potassium inside, and also to do the neurotransmission of the different types of potentials. And the lack of energy causes an accumulation of substrates, as you know, happens, for example, in asphyxia. Well, if you don't have oxygen and you are only producing 4 moles of ATP, you are not going to meet the requirements of brain in order to achieve its potential of metabolism. So you will have an alteration of the delta of intra and extracellular ion pumps. So you will get sodium inside the cell, sodium inside the neurons, with swelling of the cells, leading probably to necrosis, and you will have you will not be able to rebuild and recycle all the excitatory amino acids that you have in the synaptic cleft, and the excitatory amino acids will accumulate in the synaptic cleft and lead to a hyperexcitatory stimulus of the postsynaptic neurons, and you are going to cause the death of these neurons. So what happens when you are getting nutrients finally, and you are in the presence of oxygen? Here you have a very simple scheme of what happens. Imagine where nutrients is glucose. Glucose enters as acetyl coenzyme A into the mitochondria and goes through the Krebs cycle. The Krebs cycle is going to liberate high energy electrons that are going to be transported by specific transporters, flavin adenine dinucleotide and nicotine adenine dinucleotide. They are going to be transported to the different components of the electronic transport chain. This electronic transport chain is made up by different proteins that transfer the electrons one to the next one in an orderly manner. And in this transport from one of the components to the next one, this energy is liberated in the form of extruded protons. In the form of extruded protons, it's going to be recaptured by the 5th component of the electron transport chain, and this energy that is recaptured is going to lead to the formation of ATP. Meanwhile, every mole of oxygen is going to capture 4 electrons in the outer shell, and this is the way that oxygen neutralizes high energy electrons and impedes that these electrons are going to damage the mitochondrial structure. So what we see in the physiology, in the normal molecular metabolism of oxygen, is that we have a 4-step reduction. So the dioxygen captures 4 electrons, 1, 2, 3, and 4, and by doing so, it completes its outer shell and combined with 2 protons give rise to water, and this is a normal transitional metabolic reaction that doesn't produce any harm to our organism. However, under physiologic conditions, 2% of the oxygen is not going to be completely reducted. And under pathologic conditions such as hyperoxia or hypoxia reoxygenation, the percentage of oxygen that is not completely reduced can be much bigger, 10, 20, 30%. What happens when you are only reducing stepwise oxygen? If oxygen is reduced only by 1 electron, then you are going to have the generation of superoxide. Superoxide is a free radical, a reactive oxygen species that tends to combine with nitric oxide, giving rise to a very, very aggressive molecule that is peroxynitride. This molecule is going to damage cellular structures. However, under normal conditions, we will have superoxide dismutase, and superoxide dismutase is going to reduce superoxide with another electron into hydrogen peroxide. Hydrogen peroxide is a reactive oxygen species, but it's not a free radical. It's not going to cause any harm directly. However, hydrogen peroxide is a signaling molecule that is going to regulate many functions of the cell. But in the presence of free transition metals like iron, copper, manganese, it's going to give rise to Vento chemistry. And Vento chemistry in Haber-Weiss reaction are going to give rise to the formation of hydroxyl radical, which is the most aggressive free radical that we have in our organism, and it's going to cause a lot of damage to the nucleus, to DNA, to proteins, enzymatic proteins, structural proteins, to lipids, and also to carbohydrates. Moreover, hydrogen peroxide can also get reduced by a 3rd electron and give rise to hydroxyl radicals. So you can get hydroxyl radicals by 2 ways. And finally, if you reduce oxygen with 4 molecules, as I told you, of 4 electrons, excuse me, as I told you at the beginning, you are going to have water, and this is a neutral reaction. So what do we have? So you have that under normal conditions, 2% of oxygen is going to give rise to free radicals, and under pathologic conditions, this percentage can go up until 10, 20, or 30%. And these free radicals are going to damage DNA, causing mutations, and mutations, some of them can perpetuate, especially because preterm infants don't have the mature repair systems, DNA repair systems, the glycosylases, which repair DNA, are less expressed in preterm babies than in term babies or in adults. It's going to cause damage to the enzymes in the cells and also to the structural proteins, altering their function and their structure. And it's going to give damage and cause damage to lipids, especially those lipids who are in the membranes. You know that membranes of the cells have a bilayer of lipids, And if you destroy the lipids, you are destroying also all the receptors that are the hundreds and hundreds of receptors that have the surface of the cells, and then all the function of the cells get altered, and it's going to cause the inactivation of the cell. Here we have recently done a series of studies with a mice model of pregnant mice who gave birth to small offsprings that were born at 14% or at 21%. And when we gave these mice, we put them in 100% oxygen. Those mice that had been in 14%, so we had done a preconditioning in hypoxia, were capable of resisting better the burst of 100% oxygen. You see here, this is a control, 21%, and here you have a hyperoxic, completely destroyed mitochondria. So this is very important that you remember that preconditioning is a good measure to avoid damage to mitochondria. In order to avoid the damage that free radicals cause in our organism, we have non-enzymatic antioxidants. We have, most of them are low molecular mass antioxidants, and the most relevant is glutathione, the first one. Reduced glutathione is going to give 2 electrons to oxygen and neutralize it. But we have also others like thioredoxin, ascorbic acid, and vitamin A, uric acid, or cysteine and cysteine. We have also the possibilities of sequestration of transition metals in order to avoid the Fenton reaction. You remember that I told you that in the presence of iron or manganese or zinc or selenium, these metals are going to stimulate the creation of free radicals. If we sequester these transition metals with, for example, ferritin or transferrin or bilirubin or celluroplasmin, then we are going to impede that these metals are free and that they induce free radicals. And by physical quenching, which is the cause of vitamin A. But we have also enzymatic antioxidants. Enzymatic antioxidants, the most relevant or better known are superoxide dismutases. We have one which is in the cytoplasm, and we have also another one, manganese superoxide dismutase, which is in the nucleus, in the mitochondria, and then we have extracellular superoxide dismutase. But we have also others like catalyses, glutathione peroxidases, peroxyredoxin and glutaredoxin, which are going to reduce hydrogen peroxide. You see that when we are in a situation where oxidative stress is increasing, you see here that this mitochondria, because we are in a hyperoxic condition or we are in hypoxia, but we are recovering this baby in the delivery room with high oxygen concentrations, is going to liberate these free radicals. When the amount of free radicals increases, KEAP1 liberates NRF2. NRF2 is going to translocate into the nucleus and get binded to DNA, to a very specific region. The specific region is the antioxidant responsive element. So when they both bind, then there is an activation of the genes that express all these antioxidant enzymes that I told you before, and these are going to act upon the mitochondria and neutralize the production of free radicals. What we are seeking is a balance between reducing state, where GSH, thioredoxin, and cysteine are highly concentrated, against the oxidating state where oxidated glutathione, oxidated thioredoxin, and cysteine are predominant. So in this balance is how life usually evolves. And this balance is necessary for cell to develop and to have a normal metabolism, to divide and to get divided and to reproduce. However, in the preterm babies, due to several factors, like is the immaturity of the lungs that are necessary to give oxygen, the immaturity of the repair mechanisms of DNA, the immaturity of the antioxidant enzyme system, all these mechanisms are going to make that preterm babies, especially very preterm babies, are preterm to be in an oxidizing state. So they are going to have more facility to have DNA damage, proteins, enzymes, cell membranes, etc. So how is oxygen in the fetal life? Well, if you see this study by Dr. Snyder, that he measured the intervillous oxygen tension. You know that through the intervillous space is where the mother shares the oxygen with the fetus. So there is a passage of oxygen without any receptors or without any transporters, just by the difference of concentration. And depending on the amount of oxygen that the maternal side has, the baby is going to have a higher or a lower oxygen concentration. You see here how it happens. You see here that during the first 8 to 10 weeks in the embryological state, the partial pressure of oxygen is relatively low, between 20 and 30 millimeters of mercury. If you would increase the oxygen content in this space, in this period, to 60, 70, or 80, then you would have probably abortions. Afterwards, in the second stage, between the 10 and 12 or 14th week, when the placentation starts to occur, suddenly the amount of oxygen increases. And at the end of pregnancy, when the placenta is mature, the oxygen reaches around 50 to 60 as a median. And this is the amount of oxygen that is in the fetus at the end of gestation. You will ask yourself, how, having such a low oxygen concentration, the fetus can grow so rapidly and the fetus can have their tissues adequately oxygenated? Well, the first thing is that fetal hemoglobin has more affinity for oxygen than adult type of hemoglobin. And it liberates better, with a better pace, oxygen in the periphery than does adult hemoglobin. And moreover, the combined ventricular output of the fetus is 400 milliliters per kilogram per minute, which is substantially greater than the adult ventricular output. The fetus has a much higher hemoglobin content, and they have 100% fetal hemoglobin. For these reasons is why fetus, despite having a lower oxygen content in the blood, they are able to grow and to have a normal metabolism. However, what happens if you administer oxygen to the mother? Well, you see here fetal PO2, and you see here maternal arterial PO2. When obstetricians give oxygen to the mother in C-sections, or just because they believe that giving oxygen, it will improve the oxygenation state of the fetus, you are increasing the amount of oxygen in the arterial and in the umbilical vein. Moreover, if you measure malondialdehyde, which is derived from the peroxidation of lipids in the mother, you will see that when you increase oxygenation, you are going to increase the amount of malondialdehyde. You are going to cause an oxidative stress to the mother. But moreover, if we measure 8 isoprostanes, which are peroxidative derivatives of lipid peroxidation in the fetus, you see that as maternal arterial PO2 increases, you will have more and more amounts, which is you see it's highly significant, more amount of umbilical venous vein 8 isoprostanes. So you are causing also an oxidative stress to the fetus. And if you cause an oxidative stress to the fetus, postnatal adaptation is going to be blunted. In this study that we performed with a group of obstetricians from Finland, from the University of Helsinki, what we saw is that when the fetus is hypoxic, and we are talking now about hypoxia, as happens in diabetic pregnancies, we see that when the fetus gets more and more hypoxic, the amount of EPO, erythropoietin, in the amniotic fluid increases exponentially. So we can see, we can monitor the status of hypoxia in the fetus by doing determinations of erythropoietin in the amniotic fluid. However, what hasn't been done before, and we did for the first time in 2012, is that we measured the correlation between hypoxia, between the amount of EPO, you see it here in logarithmic scale, with metatyrosine. Metatyrosine is an oxidative derived from proteins, and this was a highly significant amount of metatyrosine when the fetus got more and more hypoxic. Moreover, when we analyzed 8-oxodehydroguanosine, which is a measure of damage to DNA, we saw that the more hypoxic the fetus was, the more consistent was the diabetes and the more the preeclampsia, then more damage to fetal DNA we got. This was correlated with the finding of hypoglycemia in the fetus. When the fetus was born, babies with high EPO and high oxidative damage, they had higher hypoglycemia, they were admitted more frequently in the NICU, they had more hypertrophic cardiomyopathy, and they had more hyperbilirubinemia. The second part of my presentation is going to deal with the oxygenation in the fetal to neonatal transition. Well, you have a normal delivery. We have already spoken about the low oxygen environment in utero. And abruptly, the fetus is born and suddenly he starts breathing and there is a massive increase of oxygen in the circulation. So during fetal to neonatal transition, we have an abrupt increase of oxygen, which will lead undoubtedly to the generation of oxidative stress. Anion superoxide, hydroxyl radical, nitric oxide, peroxynitrate, etc. These are one of them, but we have many others. We can have a normoxia, a normal situation. So we will have a physiologic oxidative stress, and these free radicals are going to activate metabolic pathways which are normal for the adaptation of the baby after birth. But we can have here on the left a situation of hypoxia. Profound hypoxia is going to give rise to hypoxemia and bradycardia, and this is going to lead to intraperiventricular hemorrhage or mortality. Or we can have situation of hyperoxia because the baby is resuscitated with large amounts of oxygen, and this is going to cause oxidative stress, which will have short and long-term consequences. In 2009, we published this paper in the American Journal of Respiratory and Critical Care Medicine, and we studied the activity of superoxide dismutase in very preterm babies below 28 weeks of gestation. And we saw that it was compared with normal babies born at term, preterm babies had a much lower antioxidant capacity. You see here in umbilical cord and in day one. However, babies who received antenatal steroids, as you see here in the white histogram, had a significant increase in the activity and were defended better against free radicals. And the same happens to catalase activity. So what we saw is that babies who are preterm have less antioxidant enzyme activity, but babies who are preterm but their mothers received antenatal steroids are in a better situation to fight against free radicals. And girls had a maturity that was about 10 to 12 days greater than preterm babies who were males. So we were worried about how we should administer oxygen to these babies, because we know the damage that hypoxia causes, we know the damage that hyperoxia causes, and we know that babies need some oxygen to start breathing and to have a normal metabolism. So together with other groups, we tried to put forward a nomogram. Our first study was done in 2003, and we were measuring during transition serial measurements of arterial blood in order to see the kilopascals, the partial pressure of oxygen. And as you see, in utero, we had 3 to 3.5 kilopascals, which is around 35% millimeters of mercury in arterial blood. But suddenly in transition, this partial pressure of oxygen increased exponentially and reached around 7 to 8 kilopascals, which is around 60 to 70% millimeters of mercury. And then it stayed quite stable. It plateaued during the neonatal transition. So we were speaking with the group of Dr. Peter Davis and Colin Morley in Australia that had the same thought as we had. Let's get a sufficient amount of babies that don't need resuscitation, that don't receive oxygen in the delivery room. Let's monitor minute by minute the SpO2 and the heart rate predactally, so we can measure it in brain. And let's see how these babies adapt after birth. So we gathered a total of 468 babies. 34% were preterm babies, less than 37 weeks of gestation, and 308 were term babies, 37 to 42 weeks. None of these babies received positive pressure ventilation and received oxygen. Here you have the reference ranges that were published in pediatrics and is one of the most cited papers in neonatology. And what we see is that practically preterm babies, not mostly late preterm, I would say that this nomogram was mainly done with late preterm babies, 34, 35 weeks of gestation, and term babies need approximately between 3 and 4 minutes to reach a median of 85% oxygen. Here you have the median. That doesn't mean that there are some babies who are normal that need around 8 to 9 minutes to reach the same, and other preterm babies, they need almost the same time. So this was for the first time it discovered that Apgar score at 1 minute cannot have in color 2 points, because there isn't almost no baby has a pink color when he is born, because he needs at least 2 to 3 minutes in order to get oxygenated. So the normal Apgar score should be between 8 and 9. But this was our first discovery. And then the second is that if the heart rate was doing well, if the baby had more than 100 beats per minute, and the oxygen was slowly increasing minute by minute without resuscitation, these babies didn't need to get positive pressure ventilation. Because if you did that you could give oxygen, you could cause hyperoxia, which causes damage to the baby, and you could blunt the normal physiological response of the chemoreceptors in the carotid bodies, who respond to low oxygen concentrations, activating breathing. So you would stop breathing and you would need more time to recover these babies. However, we have recently published a couple of months ago in late 2020. You have this paper in Journal of Pediatrics. What we did is we did a similar study as we did in 2010 with Jennifer Dawson. We did it in our hospital with around 300 babies, but we compared delayed versus immediate cord clamping to see if the blood that we are receiving in the first minutes after birth, when we are not clamping the cord, changes the nomogram of saturation. So we wanted to establish a new reference range for newborn babies with delayed cord clamping that was more than 60 seconds after birth. Babies were all greater or equal to 37 weeks of gestation after an uneventful pregnancy and delivery. Delayed cord clamping, the median was 110 seconds. We put the pulse oximeter in the right palm or wrist, and we monitored with 2 seconds measurements and maximum intensity, SpO2, and heart rate. And we put the baby at the end and prone position on the mother's chest to do skin-to-skin contact. Here you have the graph. And you see, you remember before, this was the graph that I showed you before in 2010. Babies needed more than 3 minutes. If you leave cord without clamping, when you permit that blood continues to flow through the cord, then you need only 1.8 minutes in order to achieve 85%. So less than 2 minutes time, you achieve the same saturation that you achieve when you are clamping the cord. So babies with delayed cord clamping achieve higher saturations of oxygen earlier. The second thing is that when you are dealing with babies with cord clamping, you have the first measurements you see in black of the heart rate are quite low, and it is not until 2 or 3 minutes, mostly 3 minutes is the median, go by that you have a stable heart rate of around 160. However, if you leave the cord patent and you receive the transfusion from your mother, then your heart rate doesn't go down. It's kept around 140, 150, but it doesn't vary. So there isn't this early in the early period after birth, babies with cord clamp, they have an empty ventricular output because they are receiving less blood for a couple of minutes, and they have to contract their heart with a low cardiac output and a low reception of blood. However, if you leave the cord patent, then you are receiving blood all the time. You are expanding your lungs, you are opening your arteries, and then the blood coming from the placenta is going directly through the lungs into the right and the left heart. And so you are pumping with full cardiac output. The third point would be oxygen supplementation in resuscitation and stabilization of newly born infants. You know this graph. In 2008, we showed that when you are ventilating asphyxiated babies, you are giving room air, you are reducing mortality significantly. So from then on, it was accepted that in 2010, the ILCOR guidelines recommended that it is best to begin with air rather than 100% oxygen. And here you have Dr. Ramji, Dr. Saugstad, and myself, a little younger than I am now, that we were the first to show this in a series of clinical studies. But what about preterm infants, and what are we studying now? Well, you can initiate ventilation in the delivery room with 21%, 30%, 60%, 100%. What is best? These studies were done by our group in the upper side. These studies were done in 2008 and 2009, and we compared 30 versus 90% oxygen as initial FiO2. And what you see is that we didn't find really differences. We saw a little difference in the group of 30% regarding SpO2 saturation, but it wasn't really striking. However, at the same time, Dr. Feiner in San Diego, in California, compared room air versus 100%. So this was 3 times more oxygen, and this was 5 times more oxygen. Babies with room air didn't really have a normal heart rate until they achieved 3 to 4 minutes. So they were bradycardic for 2-3 minutes. And this period of bradycardia is important. And the babies with room air, very preterm babies, less than 28 weeks gestation, were with hypoxemia and bradycardia for several minutes. You see here that babies didn't reach 80% until they were 6 to 7 minutes after birth. So they were around 5 minutes, the first 5 minutes, which I have recently published a paper, which are the most relevant minutes in the postnatal adaptation. They were not able to achieve this target saturation of 80 to 85. This was proven by Dr. Oa in Sydney. It was proven in a multi-center multinational trial called the Torpedo trial, in which she also compared 21 versus 100, and she monitored SpO2 in the first 10 minutes. When you see the babies, the most small babies, more premature babies, this happens the same as Dr. Feiner had said 10 years before. They were bradycardic, but they were hypoxemic almost until 7 to 8 minutes. So they didn't reach 85% until 8 to 9 minutes. This was a long period of bradycardia. Babies above 29 weeks, they were also hypoxemic, but they were less time hypoxemic and not so hypoxemic. When they did a post-hoc analysis of their results, what they saw is that babies below 28 weeks of gestation, 22% of these babies that had received room air died, compared to only 6% of babies that had received an initial 100% oxygen. Remember that the oxygen was afterwards titrated according to the response. So there was almost a 4 times more risk of dying in very preterm babies that receive an initial concentrations of 21%, because they needed more time to achieve a target saturation of 80 to 85. We did a meta-analysis to analyze these results, and what we saw is that babies less than 28 weeks of gestation, if you are starting with 30% or around 30% oxygen, which is called low oxygen, and you compare these babies with babies who are receiving 90 or 100, you don't see any differences in mortality. What is the question? The question is not that you are starting with higher or lower oxygen. Is that the question is, if you achieve a targeted saturation of 80 or 85%, with a normal heart rate of more than 100. And this is the important. However, the probability of reaching these higher saturations is better if you start with higher oxygen concentrations. We didn't find any differences in the secondary outcomes. So now comes the important. So our hypothesis was that preterm babies, regardless of starting FiO2, who did not reach an SpO2 of 85% at 5 minutes, were at higher risk of death or major intraventricular hemorrhage. This was an individual patient study. We included 8 randomized controlled trials that compared lower initial FiO2, which is less or equal to 30%, versus higher initial FiO2, which was greater than 65%. The babies were categorized according to 5 minutes SpO2 in those who met the guidelines, those who met the study SpO2, and those who overshot, who achieved more than 85%. You have to think that out of all this amount of babies, which were 706 babies studied, 50% did not meet the required SpO2, which means that half of the babies that we resuscitate below 28 weeks of gestation don't reach 85% at 5 minutes. And the factor that influences the most is the gestational age. Babies of 24, 25, 26 weeks have it much more difficult to reach this saturation, and the FiO2. Babies who are below 30% have more difficulties to reach. And when you combine both lower gestational ages with lower initial FiO2, then it's a huge amount of babies don't achieve this saturation. However, 29% of the babies overshot, which is 30%, which 1/3, which is important because damage caused by hyperoxia. And the most important factor was starting with higher oxygen. So we would recommend in very low birth weight infants, starting with FiO2s higher than usual, between 30 and 40 or 40 and 50, but especially to titrate rapidly if you don't achieve a saturation of 80 at 3 minutes and 85 at 5 minutes, and especially if this is combined with lower saturations, lower heart rates. So when we adjusted our results, we did an adjusted odds ratio for death or IVH. We saw that babies who didn't reach a heart rate of 100 beats per minute at 5 minutes died 4.2 times more frequently, and babies who didn't reach a saturation of 80% had 4.5 times more IVH. The same study, the most recent Cochrane review was in 2018, and they also found that in general, if you compare less than 0.4 versus higher than 0.4, you don't see differences in mortality. However, if you go baby by baby and you see which were more frequently having problems, these were babies that were very preterm and who started with the lowest FiO2. And this was the same study, the Cochrane review, who analyzed 2 of our studies and didn't find any differences in the Bayley-3 developmental scoring system. We published this paper in Pediatrics in 2016. And the same study was a consensus on science with treatment recommendation done by Michelle Welsford. And what she saw is that the studies with low risk of bias, we didn't see any differences between preterm with low FiO2 and preterm with high FiO2. And there was a high risk of bias mortality short-term randomized control at high risk of bias, then you see that there was also there weren't any differences. Here in the low risk of bias, which are studies that are randomized controlled trials done in university hospitals with a team of experts, you find better results or even better results even if you start with lower FiO2. So what we think now is it's not the initial FiO2, it's what I told you before. It's titration, and keeping heart rate above 100. So they concluded that short-term mortality wasn't difference, long-term mortality wasn't difference, and long-term neurodevelopmental impairment wasn't different. To end up, I will present you the BRADIPREM study that was led by Dr. Vishal Kapadia in Texas and has been published a couple of months ago in Resuscitation. And this is a multi-center trial, a multi-center, multinational, and is based on the motto that heart rate is the most vital of vital signs. And we know that heart rate and respiration are the most employed vital signs to assess response to resuscitation. And the incidence of bradycardia in preterm infants in the delivery room is unknown. We don't know. And we suppose that there is an association between bradycardia, prolonged bradycardia, and mortality and morbidity in the delivery room and in the NICU. And we also hypothesized that there was an interaction between prolonged bradycardia and low SpO2 in the delivery room. So we took a series of studies, eligible studies, and we were babies, preterm babies, and we had monitored in all these studies minute by minute heart rate, SpO2, and initial FiO2, and we also had retrieved complications, and all these babies were divided in 3 groups. Those who didn't have bradycardia, during the 10 first minutes, no registered heart rate was below 100 beats per minute. Those we considered to have transient bradycardia, heart rate was less than 100 beats per minute for less or equal to 1 minute. And we considered prolonged bradycardia, those who had a heart rate below 100 beats per minute for more or equal to 2 minutes. And we adjusted confounding variables for gestational age, gender, birth weight, antenatal steroids, etc. So out of a total of 535 patients, we had 205, 40%, who didn't show bradycardia at all. 115, 22%, had transient bradycardia. And, this is very important, 40% of these babies had a prolonged bradycardia, more than 2 minutes below 100 during the first 10 minutes after birth. And gestational age and birth weight were striking factors influencing these results. And SpO2, not reaching SpO2 of 80%, having an SpO2 of less than 80% at 5 minutes was also an important factor. So, in babies who didn't have bradycardias, mortality was 3%, IVH 3%, and death or IVH 6%. However, in babies who had prolonged bradycardia, more than 2 minutes, you had 5 times more death, 4 times more IVH, and 4 times more death or IVH, all highly significant. If you combine here, you see saturation less than 80, blue, at 5 minutes, and prolonged bradycardia, you see how the bar is the highest one, representing 18%. So we conclude that no differences in mortality and or severe or long-term consequences between using higher or lower initial FiO2. Initial. Very preterm infants in the lower group need more frequently oxygen supplementation to achieve targeted SpO2. So the important thing is if you are starting with high oxygen, let's say 70 or 80, you have to titrate down in the first minutes in order to avoid hyperoxia. But if you start with 30%, then you have to increase FiO2 in order to achieve at 3 minutes at least 80 and at 5 minutes at least 85. So titration is very important. Most studies show that preterm infants need an FiO2 of 0.4 to 0.6 at 3 to 5 minutes to achieve an SpO2 of 80 to 85. This is important too. So that's the limits. When you start up, you go down to 0.5, and when you start down, you go up to 0.52. Achieving saturations of 80 to 85% between 3 to 5 minutes and keeping heart rate above 100% beats per minute reflect a good stabilization process. Not achieving these targets predicts higher mortality and or IVH. And here you have a picture of my research group. Some people from the lab are missing, but these are more clinicians. And I would be ready to answer any of your questions in the expectancy that my presentation has been enjoyed by all of you. Thank you very much.
Thank you so much, sir. In June 2020, Professor Ola Sokstad inaugurated our Learn from the Legends webinar series with a talk on oxygenation and of the newborn. And 1 year and 4 months since then, the concepts of oxygenation do not cease to amaze us. It was an excellent journey. Thank you so much, Professor Máximo Vento. Sir, as usual, talks were so very good. Now we have a discussion section coming up. I invite two esteemed neonatologists of the country, Dr. Hariram and Dr. Nalini Kant, to moderate the discussion session. Over to you, moderators.
Thank you very much, Max, for your very elegant and very clear-cut presentation.
Thank you.
You took us through the physiology of oxygenation, oxygenation of the fetus, transition, and the newborn period. Thank you very much for your excellent presentation. Max will be happy to answer questions.
Yeah, I will be very happy to answer questions. Yeah.
We already have questions in the Q&A box, so we can start with that.
We have a question from Parbati Chaliha. Does height influence oxygenation of fetus? Say, for example, in places like Tawang, at a height of 16,000 feet above the sea level.
Yeah, yeah. Well, I was explaining how the fetus adapts to low oxygen under normal conditions, increasing the cardiac output and with fetal hemoglobin. When the pregnant woman is a high altitude, what happens is that the baby adapts itself also and has a higher amount of red cells, and it has a higher capacity of capturing the small amount of oxygen that the mother can transfer. So the only difference that we see in these cases is a smaller weight, but we don't see problems with oxygen. The problem would happen if this woman would be having the pregnancy at sea level, and she would have delivery at 5,000 meters altitude. Under these circumstances, the baby would probably have a pulmonary hypertension and would have the need for receiving oxygen.
Thank you, Max. We have a question from Mayank Priyadarshi. Is it that babies who are bradycardic with lower saturations are the most sick and therefore prone to mortality, irrespective of initial FiO2?
Yeah, I agree perfectly with you. Yeah. You have always to take into consideration that we don't exactly know what has happened in utero. We see a baby in front of us that is bradycardic and hypoxemic. As you know from the studies of TOS that were done with monkeys many, many years ago, the time of the primary apnea to the secondary apnea reflects the intensity of anoxia. So if a baby, once you start ventilating with a good technique or intubating and ventilating, you do it with a good technique and you are giving the proper amount of oxygen, if he doesn't react, probably he's in the secondary apnea. He has been a long time in anoxia, probably in utero. And this is when damage can be caused, and it's not a fault of a bad resuscitation procedure, okay? The problem is when a baby is born with a short period of anoxia, excuse me, and we don't resuscitate correctly because we don't titrate or we don't ventilate adequately. And this is, we are causing a delay in normalization, okay?
Thank you, Max. Dr. Nalini Kant, you have more questions there?
Thank you, Professor Máximo, for the presentation on clarifying so many practical issues on oxygen, its use in the early phase of units. So few of the questions I'm taking from the chat box here. A specific question came in the chat box regarding the practice of oxygenation. Is it different in delayed cord clamping or in the early clamping? If we are giving oxygen in the delayed cord clamping group, is it harmful to the brain more in comparison to the early clamping group, and is there evidence for so?
Okay, yeah, this is an interesting question because, yeah, what our study has shown is in normal babies. We have performed this study in term babies. We wanted to know what happens to the oxygenation in normal babies. And what we see is that there is a faster stabilization of oxygen and heart rate when you leave the cord patent. Okay? If you are having an asphyxiated baby, you are having an advantage is that if you oxygenate and you have a better pulmonary flow, you are going to achieve better oxygenation more rapidly. And you are going to keep the hemodynamics of the brain circulation and the general circulation under better conditions. And this is going to allow you to enhance resuscitation. However, this is all theoretical. We don't have yet enough experience, randomized controlled trials with enough experience and enough number of babies with delayed cord clamping, and we cannot compare both. But the studies that are going on with preterm babies, what they show is that at least is equal, but we cannot say that it's better yet. Although in experimental studies it seems to be better, but you know that sometimes the sheep and the men are not the same.
Thank you for clarifying the question. And I will take another question from here, the question session by Dr. Kalyan Bala. So why the babies with congenital heart disease tolerate hypoxia and low saturations well over prolonged periods? However, even short periods of hypoxia are not tolerated at all, any periods in babies of normal hearts. Why is it that?
Okay, this is a question that hasn't been answered yet, but we have published a paper one day ago, in which we did metabolomics of babies that were born with transposition of the great arteries, with profound hypoxemia, and when they were submitted to atrial septostomy, balloon atrial septostomy, in order to mix the blood and increase SpO2. And we did metabolomics serial metabolomics afterwards. what happens is that mitochondria in these babies are adapted to low oxygen concentrations, and they are storing the synthesis of proteins. So all the energy goes into keeping the cells alive, but there is little synthesis of proteins. Once they do the septostomy, all the metabolism switches into a normal metabolism. And then the babies start synthesizing proteins and, behaving like a normal baby. If you would go back to 70% saturation in these babies that have already done the switch, they would die. They cannot go back to the previous situation. Okay? I hope I have explained.
Thank you. Thank you for explaining things with clarity. So just in extension of this question, again, I'm taking the privilege to ask a question. Dr. Eugene's question here, Professor, whatever the fetus and neonates with the cyanotic congenital heart disease, how the oxygen target should be modified in the first few minutes of life during resuscitation, those cyanotic heart disease, if we know before?
Yeah. Well, this is very difficult to answer. I cannot give you. I would specially take care of the heart rate, and second, of the neurological response. If the babies are active, are crying, are moving, have a good tone and a good response, although they are with low saturation, I would not try to give much more oxygen, because you are not going to do any benefit, because if the heart has a congenital malformation, despite all the oxygen you give, you don't increase SpO2, and you are causing damage to the lungs and to other structures by giving too much oxygen.
Thank you again. But another question comes about therapeutics here by Dr. Rajat Atreya. The scope of antioxidant therapy, if at all we can reliably measure oxygen free radicals, will those be something that need to be looked at? Scope of antioxidant therapy.
Yeah, the problem we have had until now with the determination of free radical is that if you want to have reliable data, you need a complex system, which is mass spectrometry coupled to liquid chromatography, gas chromatography, capillary electrophoresis. So it's only complex devices that need to be managed by experts. I have 3 chemists in my group, so it's not applicable to the clinical setting. However, once we have detected the metabolites that are more relevant and more informative, our next step will be to do an ELISA method, or a simple method that allows the clinician, like you do the glucose test, to do this test with a simple method. This will be the next. But we are trying to identify because we have hundreds of metabolites, which metabolites are the most relevant ones, and which metabolites really reflect what's going on, and have a clinical value. Once we have done this, then the next step is to do a simple clinical method that allows you, when you are ventilating with a ventilator, giving oxygen, or you are doing nitric oxide, or anything that you are doing, you can monitor this as you monitor glucose, pH, or other things.
Thank you, Professor. I will request our colleague, Dr. Hariram, to go take further few questions and continue the process.
Max, I have a question. Are there any studies on cerebral oxygenation in the transition period with reference to hypoxia and hyperoxia?
Yeah, we have the group of Dr. Urlesberger in Austria has published a lot of papers using NIRS, near infrared spectroscopy, and he has done also a nomogram of how NIRS, of how cerebral oxygenation and the fraction of extraction of oxygen behave in the fetal to neonatal transition in term and preterm babies. Yeah, And we are doing it also now. We are also incorporating NIRS to our studies in the delivery room.
Thank you, Max. We have a question from Dr. Amish Vora. What is your practice in your unit? How much FiO2 do you give in babies who are term, preterm, or extremely preterm? How much do you start with?
Yeah, in term and late preterm, we start with 21. In between 28 and 32 weeks, we start with 30. And below 28 weeks, we start with 40.
Another question from Livert Mamani. What results can resuscitation have with the cord intact, and with what FiO2 would it start in these cases? I think you already answered this.
Yeah.
In your lecture.
Yeah.
Any more questions, Dr. Nalini Khan?
Yeah, it's an interesting question about a subgroup of babies. IUGR with prematurity makes it more vulnerable to the initial gradient hypoxia. And particularly in those groups, what should be best practice to resuscitate them by Dr. Ashish Jain? So it's an interesting question in particular this subset.
Yeah, I don't think in these babies the technique of resuscitation changes. We should be aware in these babies that perhaps we have to change our strategy more rapidly than in the other babies, because the response probably will be worse if we don't change the strategy. For example, regarding oxygen, perhaps in a preterm baby that it's 32 weeks, you start with 30%, you go up to 40, then the baby reaches a plateau, good heart rate, and then goes, you go down to 30, and perhaps you can go out of the delivery room with 21, 25 or so. The same weight for an undernourished baby, probably you will have to go up to 50 to stabilize him, and to keep 50 for more time. So you have to act depending on the response of the patient. But being undernourished, what tells you is that you have to be very careful, and perhaps leave this baby to the more experienced resuscitator, because titrating in the delivery room is not easy. And these babies are more prone to have complications. So I wouldn't say that if you are undernourished, you start with higher or with lower. It's that you have to adjust your resuscitation more rapidly.
We have a practical question from Dr. Venugopal. How quickly do we escalate oxygen concentration to achieve the target saturation of 85% plus?
Yeah, let's say, yeah, well, there are so many possibilities that I have to put always an example. Let's say that you have a baby who is 28 weeks and you start with 30%. And you start resuscitation. When the baby is 2 minutes, usually you should have information of SpO2. At least 2 minutes in a delivery room where you have a good equipment and where you have trained personnel. More than 2 minutes, you shouldn't wait to have SpO2. And your SpO2 at 2 minutes is 70, 75. You are sure that you are not going to reach 80% at 3 minutes. Then you should go into 50 or 60%. So you should increase in doubling the amount of oxygen, and then see how the baby responds. Okay, so the question would be answered saying that once you have the SpO2 and the heart rate monitored, you should expect in 1-2 minutes to double the FiO2 if you don't achieve a good response.
Infants of diabetic mothers already have some degree of polycythemia. In case delayed cord clamping is followed, what will be the impact on saturation, and how do you titrate oxygen under those circumstances? If you
have a polycythemia, then you have more red cells, and you don't have to care that much about having higher saturations, because if you are transporting enough oxygen, then the saturation is not that important. You should be aware of that. You don't need, you perhaps between 80 and 85, if you have a polycythemia of 60% hematocrit, then perhaps with 80% you are giving enough oxygen.
Thank you, Max. Any more
I
questions?
hate to interrupt. Can I ask Professor Max, how long can we go on? Because I know you are tied up with another event, so you have to tell us, because the questions are too many. So you have to tell us when to stop.
Yeah, you go ahead and I tell you.
Okay, thank you. Please carry on.
One clinical side from clinical question from my side. During presentation, I learned that the Apgar score in the first 1 minute may be different. It's not 10, it may be 8 or 9, because no babies by 1 to 2 minutes are pink. So do we have any clinical implication for that? If the normal low Apgar should be 8 or 9 should be normal.
Yeah, I think that when I see 10 in an Apgar score, I don't believe it. I mean, the Apgar score was, as you showed, there was this very interesting study done by Dr. O'Donnell, in which he showed videos of babies with different colors, and experts should say, what saturation do you think he has, or what Apgar score? And there was a lot of people that committed errors when interpreting the color of the baby. It doesn't matter if the baby has 8 or 10, this is normal. What you should avoid is to give oxygen because you want the baby to be pink and show the parents a pink baby. And sometimes, you know, that especially in the private practice, babies are, you know, given oxygen, so they look pink, and the parents like it better. So what I think is that 1 minute Apgar score, the most relevant data in 1 minute is heart rate. If the heart rate is okay, then all the other things can be solved. But if the heart rate is you know, is very bradycardic, then you are in trouble.
We have a question from Dr. Satya Prakash. Why does the neonate require a significantly higher SpO2 as compared to the fetus, even at 10 minutes of life?
Well, I explain you, because the fetus receives the oxygen through the intervillous space from the mother. So the intervillous space gets the oxygen from the umbilical artery. So there is, as long when you go more and more away from the arteries, the partial pressure of oxygen gets reduced. So you have in your cells, you have a 1% saturation. And so when you are having a umbilical artery has a partial pressure, a saturation of 99. But then you get to the intervillous space, to the maternal side, you have 60. So the baby gets only 50 or 40, okay? And if you go farther and farther, every time less oxygen reaches the farthest places away from the umbilical artery. So that's why the oxygen in the fetus is lower than in the mother.
Thank you, Max. Any more questions you have, Dr. Nalini Khan?
So
Have we come to the end of the question session?
we are in end of the question session. So probably we'll answer all the questions. Now, thank you, Professor Máximo, for answering patiently after the presentations. One, can I ask a just message what I understood presentation, just for our clarity in understanding? We, from the NRP guidelines, less than 35 weeks, the guidelines say 21 to 30, but with a presentation clearly says here less than 28 weeks, the mortality higher. So can I take a message from here? If a bradycardic baby of less than 28 weeks, better to start 30 rather than 21. If it's stable, less than 21, start 21, titrate upwards to 30. Can I get that clear message in the presentation?
Yeah, I have a clear message. And in babies of 24, 25, 20, 26 weeks, if they are correctly ventilated, because one thing is that you think that he needs more oxygen, and the problem is that the mask is not correctly in position. So this is something before when we have done some studies with a respiratory function monitor in the delivery room, and sometimes just by moving a little bit the mask, everything gets normal. So it was a malposition of the mask, what happened. But if you are doing things correctly, or the baby is correctly intubated, yeah, the lower the gestational age, the higher the initial FiO2 should be, and afterwards rapidly titrating, okay?
Thank you. Thank you very much.
Thank you, Max.
Thank you very much.
So it was my pleasure to be with all of you in the distance.
So,
I hope to see you soon again.
yes, Professor Max.
Face to face, face to face.
Face to face, hopefully in March when we have the physical conference. Thank you so much for confirming your presence, travel regulations permitting. And we hope to meet you in person to share, learn more from you. So it was a wonderful talk of yours. As usual, words don't suffice. The amount of information that has been conveyed with very clear messages, right from the oxygenation targets that we should be targeting, and what should be the philosophy behind targeting those values, and on to the going on to the metabolomics and things like that. So thank you so much, Professor Max. It was so kind of you to spend so much of your valuable time for us today. We are truly grateful to you.
Thank you all. Thank you very much. It was my pleasure. Bye-bye.
And at the same time, I would also like to thank two other eminent neonatologists, Dr. Hariram and Dr. Nalinikant, who have moderated today's session so well. Thank you so much, both of you. Thank you so much.
Manoj. Thank you, Max.
And
thank you all.
At the end, before we conclude, I had two very pleasant tasks. One is to thank all of you. We have quite a large number of attendees from all over the globe attending the session. So we are really thankful to all of you for encouraging us with the session, supporting us. Right now, it's almost nearing 1.5 years of this series, and you are with us all throughout, and then watching in various platforms like Zoom, YouTube, Facebook, and all that. So it is so nice of you to be with us. We request all of you to continue to support us, and let's have a journey of education together, and join us if possible physically in the physical conference, IAP Neocon National IAP Neocon, scheduled from 10th to 13th March 2022. At the same time, I would also invite you all for the next session in our series. That is on a totally different topic. That's on nutrition, and we have another legend, Professor Neena Modi from Imperial College London, who is going to take us through some very basic concepts on to the recent advances in feeding a preterm baby. So until then, thank you so much. Good morning, good afternoon, good evening, good night.